We're going to try to learn about cells and DNA and RNA, protein, and genetic engineering. Those four things are the main concepts we're going to look at, because those concepts are pretty much the concepts you need to know if you want to do genetic engineering, molecular biology in the world today. So like I said, the goal is to take you from absolutely no knowledge, we're just pretending that you have no knowledge whether you do or not, and go all the way to doing CRISPR experiments and genetic engineering experiments. Now, the book we're going to use is Molecular Biology of the Cell, and you could download a free copy. You can find that on Google Drive. It should be on the classroom and everything like that. If you scroll down to the bottom of the syllabus, it gives you a list of the chapters in molecular biology of the cell that you should read during different time periods of the class. Now, part of the course that is interesting that we're trying to do is we're trying to get you to read scientific papers. Experience reading scientific papers is really important, because if you plan on sticking with this stuff and doing it more, you're going to have to read scientific papers. You read scientific papers, and as you go through slowly, and just like look up some of the words and things that you might not know or understand, right? You're talking, when you're learning genetic engineering and biology, you're learning a whole new language. You're learning all these different words and things that mean something, and you need to figure out what they all mean. So just go through and look up all the definitions of words that you don't know. Like, that is a huge start. We divide the experiments up into weeks, but don't feel like you can't skip a week and remake up the experiments. Some of the experiments for some weeks might be pretty short and just take you like an hour or two or something like that, and others might take much longer. So, don't be afraid to start some experiments and stop them. Usually there's a lot of stopping points in experiments. Week 1 and 2, we are going to focus on cell biology and learning some basic lab techniques. Now, some of these things, they're marked as assignments. That was just an accident. You don't have to turn anything, turn these things in. So each week we should have or every week or two we have these read me firsts. So make sure you obviously read those first. They'll give you an idea of what's going on, what you should know, what you should be prepared for, what papers you should read, and stuff like that. So this week we're going to learn how to use pipette and scale and make plates and sterile technique. In the next 2 weeks, do some cultures and just learn how to work with basic molecular biology lab supplies. And make sure you do this stuff thoroughly. While pipetting might seem simple and easy, and people underestimate it, it's probably the largest place where people make errors. Professionals and people just starting alike. Like, you make errors doing pipetting, not paying attention to your pipetting, not being good at pipetting. So what you want to do is you want to make sure that 100% you know how to pipette, and 100% you're always looking at it, focusing on it, making sure you're doing it correctly, right? Because if you add the wrong amount of liquid or solution to your experiment, obviously it's going to go wrong. If your equipment's not working, obviously it's going to go wrong. So you need to make sure your equipment and you are using it correctly first before anything happens, right? We have 2 papers, and they're both related to cell biology. One is on how to create embryonic stem cells, which is a really cool paper. And it just discusses a little bit about genetic engineering and how you can genetically modify an adult cell and turn it into a a pluripotent stem cell. Pretty cool. It's something that hopefully will capture your imagination. And the other paper is about the creation of a bacterial cell where the whole genome was made using DNA synthesis rather than coming from another bacteria. So that was a pretty epic paper when it came out, and people were really impressed. Now they're trying to make synthetic genomes for yeast and even humans, hopefully in the next few years. You know, things are moving along with that stuff, but it's really interesting just to try to imagine what we can do and what can happen. There are a few things that you need to remember when we're talking about cells. The first is that as far as we as humans have figured out, cells only come from other cells. Now, there's been some work by people to try to create synthetic cells, cells from scratch, and all this other stuff. But cells, all the cells that we know came from other cells, which, you know, for me, every time I think about it and talk about it, kind of blows my mind, right? That, like, that's all we know is cells from other cells. We don't know cells that have been made from scratch. I thought we were a lot further along technologically. But yeah, it's pretty cool though, right? Because cells are self-replicating matter. So when you genetically engineer cells, what you're basically doing is you're genetically modifying self-replicating matter, right? They can grow into stuff and build stuff and create stuff. So it's something that is pretty cool, at least to me. Cells come in all different shapes and sizes. So when people are talking about cells, it's very, like, you need to know what type of cell people are talking about, right? Bacterial cells can be, and usually are, extremely tiny, right? And are generally hard to see under a light microscope, or just like, you know, a normal microscope that people would use every day. Yeast are a little bit bigger. Plant cells have different cell walls and different organelles, right? Human cells and mammalian cells are really big, bigger than both yeast and bacterial cells, and different than plant cells also, right? So when you're thinking about genetically modifying an organism, the type of cell matters, right? It's going to be a lot easier to genetically modify a cell like a bacterial cell because its cell walls are more permeable, you can get stuff in easier. Whereas a plant cell, you know, that has, like lignin and cellulose around the outside, it's a lot more difficult to get stuff inside the cell. So they use techniques like gene gun or agrobacterium to get DNA inside the cell. Plants are a little bit harder to genetically modify, but definitely not impossible with a DIY method or somebody in a home lab. It's something that you know, you can learn with practice and over time. All cells, I have written down, all cells of DNA and replicate their DNA in the same way. And I don't know if that's true, and I'm trying to, you know, prove myself wrong. But I can't think of anything at the moment. But yeah, so all organisms that are living, so excludes viruses, technically let's not count those as living right now, have DNA, and they replicate their DNA, and replicating the DNA is how they replicate themselves, right? and the DNA is what they use to become alive. So DNA, as they always tell us, is the building block of life. I don't know what that means. Don't ask me. But it sounds important. It sounds very important. So DNA isn't actually the thing that does anything, though, right? So we always talk about DNA and how awesome DNA is and what DNA does and all these things, but it's not actually DNA that does anything. The DNA is turned into RNA and then is turned into proteins. And proteins are the important part in cells. They are what make cells, alive, so to speak. They're literally like these little nano machines that can move around and do stuff. So like the way you move your muscles, the reason your heart beats, the reason that your brain works, it's all because of proteins. So proteins are probably more the building block of life than DNA, I guess. But proteins are the important thing. So when you're trying to genetically engineer something, you're trying to put DNA in the organism, but the only reason you're trying to put DNA in there is because you want to make some protein usually. Sometimes you want to make RNA or something like that. But generally, let's just say, for most cases, you're going to want to make protein. What does that protein do? Maybe that protein replaces a disease gene, right? So in like cystic fibrosis, there's a mutation in the gene. Maybe you want to replace that gene, so you have DNA make that protein. And muscular dystrophy, you have the dystrophin gene and things that are messed up, and so maybe you just want to put in a new copy of that gene to make it function better. Or maybe you want to add in some new genes that do something completely different, right? So, but what you're trying to do is you're trying to put in these genes to make new proteins to make cool things. Or maybe not cool things. I don't know. Maybe you're really boring. Diatoms? Oh man, Ted's really trying to throw out the tough questions early. How do diatoms replicate? Oh no, diatoms reproduce by binary fission. So yeah, it's this similar to like bacteria, things like that. So proteins, we're going to learn about proteins. Understand that like the amount of DNA in an organism isn't related to its complexity. So genome size is not related to the sophistication of an organism. Sometimes it's easy to get caught up in that because you could be like, well, most bacteria have tiny genomes and humans have big genomes. But despite that easy to pick out numbers, like plants will have way larger genome sizes than humans. And I think if you look at the list of genome sizes, there's a lot of cases where there's non-animal that has genome sizes bigger than animals. So don't get caught up too much in genome size and genes and things like that. It's DNA is important only in the fact that, let's say, it makes proteins pretty much. There's a lot of other stuff in DNA that's useful, but generally the important part of DNA is the protein making and what those proteins do, whether they turn you into a plant or a human. I think we might have a paper when we get to the DNA section that talks a little bit more about this, but model organisms are bullshit. So when you hear about people trying to use mice, to test stuff for humans or things like that, you have to understand, even though the DNA might be just a little bit different, it can cause huge differences in terms of immunology, metabolism, all these things. When people say that they made like a Drosophila fruit fly live longer and they try to relate it to humans, don't believe them. Don't trust them at all. Whenever anybody does an experiment in mice or not in humans and says it's related to humans, don't trust them. Immediately think they're skeptical, right? Immediately be skeptical of them, I mean. Even though DNAs might be slightly different or be similar in size and all this stuff, they're not the same. Be skeptical, right? That's one of the best qualities of a scientist. Be skeptical. Not about everything, but, try to be skeptical about, things that aren't, or people are trying to extrapolate, right? Especially when they're trying to extrapolate medical data or some cool thing from mice or worms into humans. Now, when you think of DNA and you think of proteins and you think of all these things, now basically what they are is they're just little molecules, and a molecule is just composed of atoms. And what does an atom look like? I don't know, a cloud of electrons or something. Like, we don't really look at atoms, so it's something that you can't really visualize. But they're representations and things we do to visualize these things. So these molecules of DNA are physical objects, but they're so microscopic that it's hard for us to conceptualize them. But these things are composed mostly of just 5 different things: hydrogen, carbon, nitrogen, oxygen, and phosphorus. There's some other stuff like sulfur and things thrown in there, but generally these 5 atoms make up the large majority of things in cells. So if you want to grow cells of any type, whether it's yeast or bacteria or plants or human cells, basically if you provide them with these 5 things, there's a good chance they're going to grow, or at least survive in some way. So when you're growing your cells, when you're growing things on agar plates, when you're growing things in media or whatever, understand that these media is basically, that's all it is. You're feeding these organisms sugar and some phosphorus and some nitrogen, and that's pretty much it, right? So the main component of a lot of these medias is things like yeast extract, and yeast extract is exactly like it sounds. It's basically just some yeast that everything has been extracted from them and provided to you so that you can, you can use all the good parts of the yeast that kept them alive for your organism. So the different proteins in a cell will determine how specifically that cell functions. So if you start, let's say we start with like a stem cell, totipotent stem cell in an embryo that can become anything, right? Then what happens is, as this cell gets subjected to other proteins or molecules, it starts to differentiate. And this differentiation causes different amount of proteins to be in the membrane, in the cytoplasm. It causes the shape of the cell to start to differentiate. And basically the proteins, all cells generally will have the same genes, but the proteins inside the cell and the proteins that are regulating those genes will determine the cell's specific function. H.B. Waller, how do papers on model organisms pass through if they're bullshit? Who are they convincing? Well, so you have to understand that when people are just publishing something, they're just publishing a paper, right? So generally they're not making grandiose claims. It's usually when the media gets a hold of it and they see this paper that they like cured cancer. I mean, like, they've cured cancer in mice at least 10,000 times, right? They've cured every type of cancer in mice. So it's not necessarily that the papers are bullshit. It's that the way they're portrayed is bullshit, right? It's when scientists look at the paper, generally they look at it with skepticism. They understand the mechanism and the function behind this. They understand the limits of it. A lot of people don't understand the limits of how how a mice relates to a human or how a fruit fly relates to a human. They think that these things can just be interchanged, but they really can't, right? Mr. Metric, in this class, do you teach enough info to make bacteria manufacture custom proteins? Something other than GFP? I mean, you can use the knowledge from this class to make bacteria make custom proteins, but we don't go through exactly how to do that, right? Like, all you have to do is take a plasmid you want and put it in bacteria or yeast the same way you learned in the class, but we don't teach you how to manufacture that custom plasmid. In the 102 class, it's way more in depth and it requires other equipment like centrifuge and things like that. So in the 102 class, we go over that stuff, but in 101, we're just teaching you how to how all this works, right? So how to actually genetically modify the organisms. Like there's plenty of information you'll learn that in this class that putting in more is will be overkill. Does epigenetics play a role in the expression of genes? Ted, yes, it does. What way, how, and what? That's complicated and not exactly understood, but some people say certain epigenetic things silence genes, but not all genes that have different epigenetic markers are silenced or have different expression levels. It's complicated. Epigenetics is kind of still magic. How does one cell become a toenail and the cell right next to it become flesh? Ooh, so this, Dixon's, is like this is developmental biology it gets into. And basically, it's this complex symphony, I guess, so to speak, of cells in proximity to each other, interacting with each other and secreting certain molecules and factors that tell each other what to become, right? So you can imagine if, say, you were a tile on the floor and you had instructions that said, if how many are there? There's 1, 2, 3, 4, 5, 6, 7, 8. So if you have a 3 by 3 grid of tiles, and you're the one in the center, if you have, say, 5 of the other tiles that are a certain cell type, it says, well, you should become that cell type, right? Something like that. That's not exactly how it happens. Obviously, it's more complicated than that. It depends on the tissue and all these other things, and I don't even know how every tissue becomes everything, but if you want to look up developmental biology and look up the way these things go, they have it pretty well mapped out in some model organisms like Drosophila and C. elegans. Is there a better way to go than animal models and straight up trying something in humans? Not necessarily, but I think we right now fail to use larger mammals. We should be using more larger mammals in our research, or animals that are more close to humans. The thing is that things start to get really into ethics and ethical and expensive and what it costs to keep these animals and work with these animals and use these animals. So science is this fancy dance between how much money you want to spend, how much we know about that particular organism, and how much effort you want to put in, right? So if you're using a large mammal, say like a dog or a pig or a horse or a goat or something like that, generally there's a lot less information than you'd find in, say, mice, right? So what that does is it forces people to, repeat the cycle and constantly use mice, because mice is where all the information is. Ted, there's not much difference between metabolic pathway editing and gene editing. Metabolic pathway editing is just a fancy phrase people use to try to, get more money for their companies. But all metabolic pathway editing is say putting in more than one gene that would function together synergistically to make something happen inside a cell, right? So it's generally not any more or less complicated. Now, if you want to devise novel metabolic pathways, that can start to get complicated. Or if you want to take a pathway from another organism and, make, say, THC in bacteria or something, that could be a little complicated. but generally metabolic pathways, they can be pretty straightforward or as complicated as you want, depending on how complex you're trying to do. Yeah, Sebastian, it really depends what type of cell you're trying to work with and what type of thing you're trying to get it moved towards. There are plenty of cells that move towards things. A lot of cells. I think one of the papers we might have in here is like using genetic engineering to cause human cells to move towards light, which is pretty cool. All right, any other questions before Jill? I don't know why I have these headphones on because there's like absolutely no sound. I guess you all could probably hear me even without the headphones, right? Can everybody hear me? Can people hear me? Jill, can you? Yeah, looks like the mic's working. You can hear me. For transportation of CC9 or CC13, is there better vectors or viruses? Example, using a respiratory virus versus H1 to reach particular cells, cellular DNA types. What do I prefer, genetic engineering or protein engineering? Okay, so Brandon, that I don't know anything about CC9 or CC13, so I'm not going to be able to tell you what the best vector is, and that depends on a lot, right? It depends on where it's trying to be transported to, what kind of protein it is, what it's doing. There's a lot of different things. So generally, you don't want to use different types of viruses, right? Unless you're talking about different AAV serotypes. Like, you don't just want to use, no scientist is going to use a random respiratory virus to put a gene into a human body, because these viruses have so many other things in them that could do bad things. Generally, you want to put a virus in your body that's non-replicative, so it doesn't replicate, because you can imagine if you start infecting yourself with replicating viruses, your immune system's going to freak out, and it's probably going to cause something bad. So generally people focus on AAV serotypes, adeno-associated viruses. So you should look into those and see if people have used any of those. Sudarshan, what do I prefer, genetic engineering or protein engineering? That's a tough one. Protein engineering is cool, but it generally is not as applied as genetic engineering. Genetic engineering is a lot more applied. So protein engineering is like people who like to, I don't know, write program code or write code or something like that. And genetic engineering is like people who like to build robots. So they both can be pretty cool. I'm going to go for now because Jill's going to take over. So you all be ready for Jill. She's going to show you how to do some pipetting and stuff. And we're going to switch. All right.
Okay. Hi, Peter. How's it going? Okay, so today we're going to be going over using our scales and our pipettes. And first things first is safety. So if you have long hair, you are going to want to put it up, because hair in food is gross and hair in plasmid is worse. And then next, since we're doing science, we want to put on our gloves. And this will be pretty good for everything you're going to do. You don't really need eye protection or a mask, but gloves are great. Okay, so either you already have or you are going to get a scale very much looking like this one. Pretty basic, so we just press it on, takes a second, and you want to make sure, we do everything in grams. It has different units. It doesn't like it because I'm holding it up. And right now it's going crazy because I'm messing with it. But we have different units. Make sure it's on grams up top. And then basically what you're going to do is you're going to this is all going to be on a nice stable table. You're going to put on your weigh boat and tare it. And when you do it on a table, when you press the tare button, it makes it 0 so that you don't have to subtract the weigh boat mass from whatever you're weighing out. And so it'll be like that. And when you press tare, it'll be 0. And then you just get whatever you're measuring out. And we gave you guys spoons so that you can weigh out the proper amount. And oops, left my notes over here. And so that's pretty straightforward. One note is that you want to kind of center as much as possible everything you're weighing for better accuracy. You want to bench your table that's as stable as possible, so heavier is better. If at all possible, you want your work area free of vibrations or drafts, because that will also mess up your measurements. And then ours broke off, but you all have a nice little cover for your scales. And so when you're done, you just want to clean off any of the powders that you're weighing. If it fell, that's okay. You just want to clean it off afterwards before you close it to keep your scales in good shape. And so that's basically scales. Those are pretty easy. And then we have our pipettes. So you have or are getting 3 different pipettes. I'm currently using the 100 to 1000 microliters. So you still want to use your gloves always, and you change how much the pipette holds by rotating the end part. So if I want, for example, 900 on this one, I'll just go like that, and it's at 900. Then you're going to put on your tips. So we sent you 3 different sizes of tips for your 3 different sizes of pipettes. So when you put it on, you just make sure that it's on nice and firmly. Sometimes do a tap and bring it up. Then you're going to take your liquid that you are pipetting and you want to insert the tip far enough down so that you'll make sure that you won't suck up any air bubbles, but you don't want to stick it in as far as it can go, because then you'll have a lot of liquid adhering to the outside and it could mess up your measurement. So you stick it in a good amount, and first you do 3 times where you suck up the liquid and then just put it back, because that coats the inside of your pipette tip so that you don't lose any an amount of liquid the first time you're doing it. Also, right now I'm just using colored water, so it's coming up pretty quickly. However, if you're pipetting something thicker, you might want to wait, and I generally like to count to 3 if it's a thick liquid, to make sure that it's had enough time to uptake all of the liquid. And you want to uptake and put out nice and smoothly. You don't want to flick it or anything. That's gonna mess up your measurement. When you uptake, you go down. There's 2 stops on a pipette. There's what's called the soft stop, which is where you feel slight pressure, but then you can push it down more. And the reason for that is you go to the soft stop to pull up your liquid. And then when you go down to release your liquid, you go all the way to the to the full stop, and then that makes sure that all the liquid that was in there is pushed out. It just pushes it out with some extra air. You want to have the pipette go in at 90 degrees when you're taking up. However, when you are putting it back into another container, you want to slightly angle the container so that it doesn't splash and it goes in nice and evenly. If your pipette is full, you do not want to lay it on its side because then the liquid can get into the actual pipette and contaminate it, and that would be a pain to clean, so you definitely don't want to do that. If your pipette does get dirty, you can clean it with 70 to 90% ethanol, and that'll help sterilize it so that you can use it again without contamination. Once a year, you want to calibrate your pipettes. I believe there's instructions in there on how to do it. Basically, you're using the density of water and you have a scale and you like weigh out the volume to make sure that it's accurate. And if you have more questions about that, go ahead and ask that in the Google Classroom and we can go over that a bit more. Make sure your tips, we know that the tips we sent you will fit well. However, if you order more tips and they seem to be different, just one time check and make sure that it's not leaking or anything. You'll see it's pretty readily. If you just wait and nothing comes out for like a minute or so, you're totally fine. But sometimes like I don't even like press down hard enough and they start to get drops down and I see it. So then I just toss that pipette tip and get another tip. And then to get the tip off of your plunger, because this is just water, but maybe it's contaminated with something you don't want to touch, you basically press this part, and what it does is it, put it in my little weigh boat. It takes off the tip without, oops, that fell anyway, but yeah, it takes off the tip without you having to touch the end so that you're as clean as possible.